Hello everyone, I'm Dr. Kanika. In today's session, we will cover the topics like blood volume, RBC, blood transfusion, spleen, and possibly we will cover the body fluid. So now the first topic is the blood. Blood is a connective tissue in a fluid form. It is considered as the fluid of life because carries oxygen from lungs to all parts of the body and carbon dioxide from all parts of the body to the lungs. It is also known as a fluid of growth because it carries a nutritional substance from the digestive systems and hormones from the endocrine gland to all the tissues. The blood is also called the fluid of health because it protects the body against the diseases and gets rid of waste products and unwanted substances by transporting them to the excretory organs like kidneys. Now, the properties of a blood. The color. Blood is a red in a color. Arterial blood is a scarlet red because it contains more oxygen, and the venous blood is a purple red because of the carbon dioxide concentration. Now, the volume. Average volume of the blood in a normal adult is a 5 liter. In a newborn baby, the volume is a 450 ml. It increases during the growth and reaches a 5 liter at the time of puberty. In females, it is slightly less and it is about 4.5 liter. It is about 8% of the body weight in a normal young healthy adult, weighing about 70 kilogram. Now, the pH. Blood is slightly alkaline in nature and its pH concentration is a 7.4. Then viscosity. Blood is a 5 times more viscous than a water and it is mainly due to the red blood cell and a plasma proteins. Now the compositions of a blood. Blood contain blood cells which are called the formed elements and the liquid portion known as a plasma. Again, plasma divided in many substances like a solid, water and gases. Solid contain the 7 to 8 percentage of the plasma. Water content is a 90 to 93 percentage of the plasma, and the rest are the gases. In a solid substances, there is the organic substances and other is a inorganic substances. In organic substances, there are a plasma proteins. हाँ बोल रही हूँ मैं तो हाँ बोल रही हूँ So blood also contains enzymes like amylase, carbonic anhydrase, acid phosphatase, alkaline phosphatase, lipase, esterase, protease, and transaminase. In non-protein nitrogenous substances, blood contains ammonia, creatine, creatinine, xanthine, urea, and uric acid. It also contains the antibodies and the gases like oxygen, carbon dioxide, and nitrogen. Now, the other component of the blood is the blood cells, in which three types of the cells which are present in a blood, that are red blood cells or the erythrocytes, white blood cells or the leukocytes, platelets or the thrombocytes. Now, the property like hemocritic value. If a blood is collected in a hematocrit tube along with a suitable anticoagulant and centrifugate for 30 minutes at a speed of 3000 revolutions per minute, the red blood cells settle down at the bottom, having a clear plasma at the top, and plasma forms 55%, and red blood cells form 45% of the blood. So we can say that the blood contains 55% of the plasma and 45% of the cells. Now, the other substance is the serum. Serum is the clear, straw colored fluid that oozes from the blood clot when the blood is shed or collected in a container. Okay. In this process, the fibrinogen is a converter into the fibrin, and the blood cells are trapped in the fibrin, forming the blood clot. So basically, it says about the fibrinogen form into the converted into the fibrin, and the blood cells are agglutinate in that area, and the serum is produced. After about 45 minutes, serum oozes out of the blood clot. For the clinical investigation, serum is separated from a blood cells and clotting elements by a centrifuge. Volume of the serum is almost the same as that of the plasma, which means 55%, and it is different from a plasma only by the absence of a fibrinogen. Like serum contain all the other constitution of the plasma except a fibrinogen. Fibrinogen is absent in a serum because it is converted into the fibrin during a blood clotting. So we can say that serum is equal to plasma minus fibrinogen. So serum is only a plasma content which exclude the fibrinogen. Now we are going the important about blood, which are the functions. So the number one is a nutritive function. Nutritive substances like glucose, amino acid, lipids, and vitamins derived from a digested food One second. Okay. So the transport of hormones and enzymes. Hormones which are secreted by the ductless means the endocrine glands are released directly into the blood. The blood transporter these the hormones to the target organs or the tissues. Blood also transport the enzymes. Let's say the example the hormones which created from the hypothalamus glands or the which important which transport through the blood to the particular organ for the particular action. Now the regulation of a water balance. Water content of the blood is a freely interchangeable with interstitial fluid. This helps in the regulation of a water content of the body. Now, the other function is a regulation of a acid-base balance. Plasma proteins and the hemoglobin act as a buffer and help in the regulation of the acid-base balance, which is very important to balance our acid and base concentration in blood. Then regulation of the body temperature. Because of the high specific heat of a blood, it is responsible for the maintaining the thermoregulatory mechanism in the body. Like example is the balance between the heat loss and heat gain in the body when we face the different weather and different conditions. Then it regulate our body temperature. Then the other function is a storage function. Water and some important substances like a protein, glucose, sodium, and potassium are constantly required by tissue. Blood serves as a ready made source for these substances, and these substances are taken from blood during the condition like the starvation, fluid loss, electrolyte loss, and etc. So it is mainly protect from these conditions like starvation, blood loss, electrolyte loss. So in temporary condition, they protect body. Then the important role in a defensive function. Blood plays an important role in a defense of the body and in which the white blood cells, particularly white blood cells, are responsible for the defense mechanism. Neutrophils and monocytes engulf the bacteria.
So, so in, okay. So the high altitude blood volume increase in the high altitude. It is because of the hypoxia. Okay. So we're talking about a high altitude. Blood volume increases in a high altitude. It is because of the hypoxia, which stimulate the secretion of the erythropoietin. As we know about the erythropoietin is a hormone which increase the formation of the RBCs, and it's a directly effect on the blood volume. So it induces the production of a more RBC, and which lead to the increase in the blood volume. Now, the emotions. Emotion also plays on the volume of the blood. So, excitement increase the blood volume. It is because of the sympathetic stimulations, which causes the splanchnic contractions and the release of a stored blood into the circulation. So, every emotions and excitement are stimulate our nervous system, and that's why it's also increase the blood volume. Okay. So now the pathological variation, we will see now the pathological variation. So abnormal increase in the blood volume is called the hypovolemia, and abnormal decrease in the blood volume is called hypovolemia, and we will see this pathological variation in applied physiology later on. Okay, so now the measurement of the blood volume. So blood volume is measured by the two method, like a direct method and the indirect method. Indirect method is employed only in the animals because it is involves the sacrificing the life. So we cannot do it on a human being. So it is always do on the animal, and the animal is killed by the person, and the blood is collected. So the blood vessels in which we collect this blood of the animals and the tissues are washed through the uh, with non quantity of the water or the saline, and this is added to the blood collected already. So the volume measured from this, the volume of the water and saline which we used for the washing the tissues is deducted. So easily we find the volume of the blood in the animal, and it has a historical phenomena. This method was the first employed by the Welker in the 1854, and then the same method used by the some other scientist. So this is the criminal method. So determine the blood volume. It is not, we cannot use in the human beings. We can use the indirect method, which is another method. So what we do in the indirect method. The indirect method is advantageous because it is used the measure of the blood volume in the human being without causing any discomfort or any difficulty to the subject, in which the blood volume involves the two steps, in which first step is a determination of the plasma volume, and the second is a determination of the blood cell volume. So now how we find the determination of the plasma volume. So plasma volume is determined by the two method. Then the indicator or the dilution technique, in which we inject the dye, and the other is a radioisotope method. So the in first method, we inject the dye which is used to measure the plasma volume is the Evans blue or the T1824, in which procedure we 10 ml of the blood is drawn from the subject, and this is divided into the two equal portions, and that to one part a non quantity of the dye is added, and that it is used to control samples in the procedure. The other portion is used to determine the hematocrit value. Then a known volume of the dye is injected intravenously. Then, after 10 minutes, a sample of the blood is drawn. Then another four samples of the blood are collected at the interval of a 10 minutes. So, in first, after 10 minutes, we take a first sample, and then the other four interval, like 10 minutes interval, we take a four other sample, and then all the five samples are centrifugated, and plasma is separated from the samples. In each sample, the plasma, the concentration of the dye is measured by the colorimetric method, and the adverse concentration is found. The subject's urine is collected and the amount of a dye, because the dye is also excreted by the urine, which also measured. Then there is a one formula to count the volume. So that formula is volume is equal to amount of a dye injected in the subject minus amount of excreted through the subject by average concentration of the dye in a plasma. Now the another method is which using the radioisotope method, in which iodine 131 or the iodine 132 is injected in the body. After sometimes a sample of a blood is collected, and the radioactivity is determined by using an appropriate counter. From this, the plasma volume is determined. So basically, these all techniques are used to measure the blood volume. Okay. So now the determination of the blood cell volume. In above procedures, we will find the plasma. Now, the blood cell volume. How we determine the blood cell volume? We will check it. Blood cell volume is determined by the two method, hematocrit value and the radioisotope method. The hematocrit value is a centrifuging of the blood and measuring the PCV, which is known as a packed cell volume, in which PCV is expressed in a percentage. Okay, so if this is a deducted from 100, the percentage of a plasma is known. And from this and from the volume of plasma, the amount of the total blood is calculated by the using the formula, which is blood volume is equal to 100 into amount of a plasma by 100 minus PCV. Now the another method is a radioisotope method. Volume of a blood cell is measured by the radioisotope method also. So radioactive, in which we use the chromium 52, is added with a hyperonized blood and incubated for the 2 hours at a 37 degrees Celsius temperature. During this time, all the red cells in the blood are tagged with the chromium 52. Then this is injected intravenously. After giving sufficient time for the mixing, sample of the blood is drawn, and the hematocrit value is determined by measuring the radioactivity in the blood sample. Radioactive ion, also the phosphorus, is also used for determining the hematocrit value. So if we can say that the radioactive use for the hematocrit value of the blood is a chromium phosphorus, and in blood volume we use the iodine radioactive. So this keep in mind. So now the regulation of the blood volume. So various mechanisms are involved in the regulation of the blood volume. The important ones are the renal and the hormonal mechanism. So basically, the renal means the kidney and the hormones mechanism are two major system which plays important role for the balancing our blood volume. Okay, so hypothalamus is a endocrine gland which plays a vital role in the activation of these two mechanisms during the regulation of the blood volume. So hypothalamus is the plays the major role for the balancing our blood volume. When the blood volume increase, hypothalamus causes the loss of a fluid from the body. When the blood volume reduced, hypothalamus induces the retention of water, and then the hypothalamus regulate the extracellular fluid volume and the blood volume by acting mainly through the kidneys and the sweat glands, and by inducing the thirst. So it automatically inducing the thirst. So person will go for the water, and that's why balancing the volume. Then the hormones also play the important role and involved in the regulation of the blood volume through the regulation of the ECF, extracellular fluid. So hormones which are involved in the maintenance of ECF volumes are, these hormones are very important. So you can write that antidiuretic hormone, aldosterone hormone, cortisol, and the atrial natriuretic peptides. These four plays the major important role to regulate the blood volume. So now we can moving further with the applied physiology of the blood volume. So there is a few terms like hypervolemia. So what is hypervolemia? So increase in the blood volume. Hyper means increase. So hypovolemia is the increase in the blood volume. It's known as the hypovolemia. It occurs in the following pathological conditions. So now we cover the pathological conditions which involves in the hypervolemia or the hypovolemia. So in which hypovolemia increases in the which pathological condition. First is a hyperthyroidism. Hyperthyroidism means blood volume increases because the thyroxine increases the RBC count and the plasma volume. Means hyperthyroid is the hormones, and if it increase in the blood, so it also increase the blood volume. Now the hyperaldosteronism. In hyperaldosteronism, excess retention of the sodium and water leads to the increase in the ACF volume, and that's why it also increases the blood volume. Third is the cirrhosis of the liver. It is a pathological condition related to the liver in which the blood volume is more because of the increase in the plasma volume. Number four is a congestive cardiac failure. Retention of a sodium occurs in this condition, in which when the cardiac failure occur, the sodium retention also occur, and it leads to the water retention and also increase in the ECF volume, plasma volume, and the blood volume. Okay, so these four conditions which we see in the hypovolemia, and now we see the hypovolemia. So decrease in the blood volume is called the hypovolemia. It occurs in the following pathological conditions. Number one is a hemorrhage or the blood loss due to the accident, or the acute hemorrhage occurs due to the cuts or the accidents, and the chronic. Acute means the sudden, and the chronic, which means takes the time. So acute hemorrhage due to the cuts or the accidents, and the chronic hemorrhage due to the any ulceration found in the system, or the bleeding piles, and the excessive uterine bleeding in female during menstruation, or the any other condition. So it will lead to the hemorrhage, which are acute or chronic, and ultimately lead to the hypovolemia. Then the number two is a fluid loss. So fluid loss occurs in a burns, vomiting, diarrhea, excess in sweating, and the polyuria, which means polyuria means the increases in the urine formation. In all these conditions, we can find the hypovolemia. Then hemolysis. Now, the what is a hemolysis? The excessive destructions of the RBCs, which means normally RBCs are destructed in 120 days lifespan of the RBC. Okay. So, in hemolysis, excessive destructions of the RBCs occur because of the presence of the various hemolytic agent and the other factors such as hypotonic solutions, neck venom, acidity or alkalinity, mismatched blood transfusions, hemorrhagic smallpox, and measles. These all pathological conditions lead to the destructions of the RBC, and excessive destructions of RBCs, which lead to the decrease in the blood volume, and which ultimately decrease in the our blood volume. Okay, so it will lead to the hypovolemia. Then the anemia. Anemia is also play the important role in the hypovolemia. Blood volume decreases because of the various type of the anemia, because of the decrease in the RBC count. In some cases, the quantity, which means the volume of the blood, remains the same, but the quality blood alter, which means the volume is not altered by the anemia, but in which the quality of the blood is a decrease and it ultimately lead to the hypovolemia. So blood becomes diluted, and because of the entrance of the fluid into the blood vessels, it will lead the quality of the blood. Then again, hypothyroidism. In hyperthyroidism, we will see the hyper volume, and then in this hypothyroidism, we will see the hypovolemia. During hypothyroidism, the blood volume is decreased because of the reduction in the plasma volume and the RBC count. So, we will cover in this blood volume all the conditions which are physiological, pathological, how we can measure the blood volume, the importance of the blood, and the systems which are which has effects to balance our blood volume. So now we will moving ahead with the RBC. So RBC, which means the red blood cell, are the non-nucleated. 1 minute. Non-nucleated, which means it not contain nuclear formed elements in the blood. RBC are also known as erythrocytes. Erythros means the red and cytes means the cell. So red blood cells or the erythrocytes. Red color of the red blood cells is due to the presence of the coloring pigment called the hemoglobin. So why RBCs are red? Because of the hemoglobin, which is a coloring pigment. So it gives the color red to the RBC. RBCs play a vital role in a transport of the respiratory gases. RBCs are the larger in number compared to the other two blood cells, namely the white blood cells and platelets. So basically, we found the different type of the cells in the blood, like RBCs, WBC, platelets. But the RBCs are the larger in number compared to the other two. Then the normal value of the RBC. RBC count ranges between the 4 to 5.5 million per cubic per mm of the blood. In adult male, we find the 5 million per cubic mm, and in adult female, there is a less quantity is a 4.5 million per cubic per mm. Now we see the morphology of the RBC. So in that shape, so normally the RBCs are the disc shape or the dumbbell shape or the biconcave. We use the three terminology: disc shape, biconcave, or the dumbbell shape of the RBC. The central portion is a thinner and the periphery is a thicker, so it look like a dumbbell shape. The biconcave contour of the RBCs has some mechanical and the functional advantage. Why these RBCs have this type of shape? Because it have a major role in the functional and mechanical advantage of the RBCs. So what is that advantage of the biconcave shape of RBC? We can see that biconcave shape helps in the equal and the rapid diffusion of the oxygen and the other substances into the interior of the cell. So because of this type of the shape, RBCs plays a rapid and equal diffusion of the oxygen. Then larger surface area, because of the periphery has a larger surface area. So it provides the absorptions or the removal of different substances very quickly. Then the minimal tension. Minimal tension is offered on the membrane when the volume of the cell alter. And because of this biconcave shape, while passing through the minute capillaries, RBCs squeeze through the capillaries because of the capillaries are very minute. So because of this biconcave shape, they easily move through the capillaries without getting damaged. Okay, so it is advantage of the biconcave shape of the RBC. Now the normal structure. Red blood cells are the non-nucleated. Only the mammals which has the nucleated RBC is a camel. So it is a very important. All the human beings and all not contain the nuclear in the RBC, but the only mammal which contain the nucleated RBC is a camel. Okay. So because of the absence of the nucleus in a human RBC, the DNA is also absent, because we all know the DNA is a present on the nuclear. nucleus. So in this absence of the nucleus, in the human, we don't find the DNA in the RBC. Then the other organelles such as the mitochondria, Golgi glucose uptake by this cell is not controlled by insulin. So the whenever the uh, RBCs uptake the glucose, which is not controlled by the insulin. Okay, RBC has a special type of a cytoskeleton, which is made of the actin and spectrin, both the proteins. So these are the proteins which known as actin and spectrin, which form the cytoskeleton of the RBC. Both the proteins are the anchored to transmembrane proteins by means of the another proteins called the ankyrin. So three proteins are very important: actin, spectrin, and ankyrin for the formation of the RBC. Now, absence of a spectrin result in the hereditary spherocytosis. So if someone have the absence of the spectrin, so it's known as the hereditary disease with the spherocytosis. In this condition, the cell is a deformed, loses its biconcave shape and become a globular. And the spherocytes is a very fragile and easily ruptured in a hypotonic solution. Okay, so now we will move ahead with the properties of the RBC. So now the Rolex formation. When blood is taken out from the blood vessels, the RBCs are piled up one above another like the pile of coin. If we see the pile of coin, same structure we can show on the RBC. This property of the RBC is called the Rolex formation. It is accelerated by the plasma proteins, globulin and fibrinogen. So basically, globulin and fibrinogen are plays a major important for the this property, which is known as a Rolex formation. Okay, blood RBCs have a specific gravity also. So specific gravity is a 1.092 to 1.101. Now the PCV. PCV means the packed cell volume. Black cell volume is the proportion of the blood occupied by the RBCs expressed in a percentage. So PCV is expressed in which measurement? So it's expressed in a percentage. It is also called the hematocrit value. So other name is the hematocrit value, and the packed cell volume is the same. And it is a 45% of the blood, and the plasma volume is a 55%. So we can say that the 45% of the blood and the 55 cells and the 55% is the plasma. Another property is a suspension stability. During the circulation, the RBCs remain suspended uniformly in the blood, and this property of the RBCs is called the suspension stability. Now, moving on with the lifespan of the RBC. Average lifespan of the RBC is about 120 days. After the lifetime, the senile RBC, which means the old RBCs, are destroyed in the reticuloendothelial system. After every 120 days, the old RBCs are destroyed and the new RBCs are formed, and which enter in the circulatory system. Okay. So, how determination of the lifespan of the red blood cell? Lifespan of the RBC is determined by the radioisotope method. So now, in this, RBCs are tagged with the radioactive substances like a radioactive iron or the radioactive chromium, and then we can find the lifespan of the RBC by the studying the rate of the loss of the radioactive cell from the circulation. It is a very easy method to know about the lifespan. Okay. So now the fate of the red blood cells. So when the cell become older, which is the 120 days period, the cell membrane become the more fragile of the RBC, and then the diameter of the capillary is less or equal to the that of RBC. Younger RBCs can easily pass through the capillaries, and however, because of the fragile nature, the older cells are destroyed while trying to squeeze through the capillary. So when it become older, then it not easily pass through the capillary, and it squeeze the capillaries, and then the destruction occurs mainly in the capillaries of the red pulp of the spleen. So spleen is the major organ for the destructions of the RBC. So why spleen? Because in the capillaries of the red pulp of the spleen, because the diameter of the splanchnic capillaries is very small. In the human body, the capillaries of the spleen is very small compared to the other organ. So the spleen is called the graveyards of the RBCs. So which organ is known as the graveyard of the RBC? So we can say the spleen. Okay. So destroyed RBC are fragmented, and a hemoglobin is released from the fragmented parts. Now the what happened with the destroyed RBC? Phagocyte the our this hemoglobin, and in which particularly the macrophages present in the liver, spleen, and the bone marrow. So what happened with the RBC destruction, which is destructed in the spleen, and then the hemoglobin is released from that fragmented parts, and it is phagocytes by the macrophages, and particularly macrophages present in the liver. So liver and spleen plays a major role on the in the destructions of the RBC. Okay. Now hemoglobin is degraded into the iron, globin, and the porphyrin, which all are the protein. Then what happened with these substances? The iron combined with the protein called the apoferritin to form the ferritin. So now we can say the protein which exclude from the hemoglobin, which is a porphyrin. Now the iron plus porphyrin combine with the porphyrin protein and the form the another protein which is known as the ferritin. And now this ferritin, which is stored in the body and reused later. So hemoglobin is not excreted or anything will happen with the hemoglobin. It is reused later on because the globin and and peripherin, which all are the protein of the hemoglobin, and the iron combined with this protein apoferritin and from the ferritin, and which stored and which is used later on. Now the globin, what happened with the globin? So globin enters the protein depot for the later use. Okay. And the porphyrin is degraded into the bilirubin, and which is excreted by the liver through the bile. So three type of the hormones: globin enters into the protein depot system, and the porphyrin is degraded into the bilirubin, and which excreted by liver through the bile. So this three type of the protein. Now daily 10% of the RBCs which are senile are destroyed in a normal young healthy adults. So one young healthy adults, 10% of the RBCs destroyed in every human beings, in normal human beings every day. Okay. So they will ask you how much RBCs are destroyed every day in a young healthy adult. So it is a 10% of the RBC. Okay. So it causes a release of about a 0.6 gram per deciliter of the hemoglobin into the plasma. From this, 0.9 to 1.5 milligram per deciliter bilirubin is a form. Okay. So now moving ahead with the functions of the RBC. So major function of the RBC is the transport of the respiratory gases. So how they can do this? So transport of the oxygen from the lungs to the tissue, hemoglobin in the RBC. In RBC, particularly the hemoglobin, which is a plays a major role for the transfusion of the gas. So hemoglobin combine with the oxygen and form the oxyhemoglobin. So, about a 97% of the oxygen is transported in a blood in the form of the oxyhemoglobin. Oxygen is not transport through the body, but it will join to the hemoglobin and form the oxyhemoglobin. And in form of the oxyhemoglobin, it is transported throughout the body. Okay. Number two is the transport of the carbon dioxide from the tissue to the lungs. So now hemoglobin, in which also the hemoglobin play the major role. Hemoglobin combine with the carbon dioxide and form the carb hemoglobin. About 30% of the carbon dioxide is transported through this system. So we will easily say that oxygen is transferred through the oxyhemoglobin, and the carbon which is transferred through the carb hemoglobin. And 97% of the oxygen is transferred through the oxyhemoglobin, and the 30% which is transferred through the transported through the 30% of the carbon dioxide is transported through the carb hemoglobin. Okay, so now RBCs contain a large amount of the carbonic anhydrase, which is an enzyme. This enzyme is necessary for the formation of the bicarbonate from the water and the carbon dioxide. So, water and the carbon dioxide present in the body, in which the carbonic anhydrase is work on this enzyme is work on the water and the carbon dioxide, and thus it helps to transport carbon dioxide in the form of a biocarbonate from the tissues to lungs, and about a 63% of the carbon dioxide is transported in this form. So 30% is transported through the carb hemoglobin, and the 63% is because of the carbonic anhydrase enzyme. So two carbonic anhydrase enzyme and the carb hemoglobin formation, which plays the major role for the transporting carbon dioxide. Now, the third function is the buffering action in the blood. Hemoglobin function has a good buffer. By this action, it regulates hydrogen ion concentration and thereby plays a role in the maintenance of the acid base balance. So, acid base balance in which the RBCs plays a major role as a buffering agent. Then the blood group determination. It's a major role of the RBCs to find out the blood group of the any specific person. So by how we find the blood group? RBCs carry the blood group antigens like the A antigen, B antigen, and the Rh factor. So particular each RBCs have the different type of the antigen like the A or B or the Rh factor, and by the processing we find the particular blood group of the particular person, and this helps in the determination of the blood group and enables to prevent the reactions due to the incompatibility blood transfusion. So it plays the major role during the blood transfusion. We have to find out the specific blood group of the person, and then only we can going for the process of the blood transfusion. Okay, so now variations in the number of the red blood cells. So again, there is a physiological variation and the pathological variation. So increase in the RBC count. Okay. So wherever there is increase in the RBC count is known as the polycythemia. Poly means the increase, and cythemia means the cell. So it occurs in the both physiological and the pathological condition. When it occurs in the physiological condition, it is called as the physiological polycythemia. Physiological polycythemia. Okay. The increase in the number during this condition is a marginal and temporary, and it occurs in the following condition. Okay, so age. So at birth the RBC count is 8 to 10 million per cubic mm of a blood, and the count decreases after the 10 days of the birth due to the destructions of the RBCs causing a physiological jaundice in some newborn babies. So this physiological jaundice is happen in the babies. So that's why the RBC counts is decreases after the 10 days of the birth of the baby. Okay. So, however, in infants and growing children, the cell count is more than the value in the adult. So that's why we find a more RBCs in the infants and the newborn baby. So keep remember that the babies and the infants have a more RBC value than the adult. Okay. So now sex before puberty and the after the menopause. Before puberty and the after menopause, in female, the RBC count is similar to the males. But during the reproductive period of the female, the count is less than that of the males because of the menstruation and the all. Okay, so now the high altitude. Inhabitants of the mountains above the 10,000 feet from the mean sea level, so height is at 10,000 feet, have the increased RBC count of a more than a 7 million per cubic mm. And it is due to the hypoxia. Because of the decrease in high altitude, there is a decrease in the oxygen supply to the tissue. So it will ultimately lead to the decrease in the RBC also. So hypoxia stimulate the kidney. So this type of the hypoxia which created in the high altitude, which stimulates the kidney to secrete a hormone called the erythropoietin, and this hormone, the erythropoietin, in turn, which stimulate the bone marrow to produce some more RBC. So high altitude, and we can see it in a cycle like high altitude, which lead to the hypoxia, which means a decrease in the oxygen. Then which it lead to the erythropoietin hormones stimulation, and then erythropoietin stimulate the bone marrow, and bone marrow produce more RBC, and then the balance of the RBCs occur in the high altitude position. Okay, so now the muscular exercise. So there is a temporary increase, not permanent, but temporary increase in the RBC count after the exercise. And it is because of the mild hypoxia, because if we doing exercise, there is a mild hypoxia and contraction of the spleen. Okay, so there is a muscular exercise also increase in and temporary increase in the RBC level. Okay, so we will see the other point later on.